So let's talk about the kind of things that can go in and out of a cell. There's a few things that are able to pass through the cell. It's lipid bilayer, and other things are going to have to get sent through different directions. Water can go from one side of the cell to the other really easily, and how they do that is because these lipid bilayers allow them to sort of squeeze through and move. No problem. The next thing that can go through are small non-charged molecules. So non-charged molecules can go across the membrane as well, as long as they're really small and they have really no charge on them. So like glucose and things like that can. Things that can't move through that process are going to be things like charged molecules. Charged molecules won't be able to pass through. They'll sort of bounce off and then sort of have to go away. Same thing, it won't be able to come through, it just stays inside. And then also large molecules. Large molecules will have to sort of stay on the outside or on the inside. For these guys to come through, there's going to have to be a protein, and that protein is going to be only the thing that sort of recognizes this one item and lets them in. If you remember our experiment that we did in lab, we found out that some things were able to pass through. We found out that water was able to pass through. We were able to find out small molecules that didn't have a charge that were able to pass through. But things like starch that are going to be really big and things like that albumin, they weren't able to cross that membrane. It was because they were just way too big. So we're going to talk about the kind of things that can move across the membrane and how they pass through. Because we know stuff has to go across the membrane. And there's two different types of membrane transport. There's passive energy and active. Both of these are going to be a way to move stuff across membranes. And active energy is just called active transport. Passive energy just means that it can do it without energy, but there might be different ways to do it. So let's talk about it. Diffusion, osmosis, passive transport, and then active transport are the ones that we're going to cover. Facultative diffusion is kind of in this method where they all sort of move across, but let's just talk about the simple ones. So what you're seeing here is just that things are in high concentration on one side of, let's say, a cell, or maybe even a room or whatever, and over time they're going to cross to the other side until they get to equilibrium, equal on both sides. Here it makes a little more sense because you can see two different colored balls on one side, on the other side. Over time they're going to diffuse, and eventually there'll be an equilibrium everywhere in the cell, or even outside of a cell, depending on what we're talking about. Diffusion is going to go from an area of high concentration to low. We call it down a concentration gradient because it's like going down a hill, and it's really easy to do. And because it's easy to do, there's no energy required. You don't really need a protein to help you do that. You don't really need a membrane, because with diffusion, this could be something like perfume in a room, or just even putting a little bit of sugar and water, and eventually it'll diffuse through. If we keep going, you could see that we have a situation where we put, let's say, sugar in water. The sugar would be the solute, the water would be the solvent. Over time, they will diffuse to be in equal concentrations everywhere, and that's going to be called the solution. We do simple transport all the time. One place we do it is our lungs, but this is not the only place. We do it in our kidneys, and we do it in every cell, but let's just look at this one because it's kind of easy to sort of wrap our heads around. Oxygen moves into your body, into your lungs. When it's in your lungs, there's an area of high concentration inside these little things called alveoli. They're like these little sacs. And there's a low concentration in the bloodstream. So what's going to happen is the oxygen is going to move from high to low and move into the blood. Eventually, if the blood just sat here and it wasn't rushing, all the time past this, it would get to equilibrium. But what's happening is it's always low here, it's always high here, so it's going to move through. If we look at the opposite of what's happening here, in the blood, there's going to be a high amount of CO2, and here there's a low amount of CO2. So the CO2 will go ahead and go into the alveoli, and then eventually out of your mouth. Osmosis. Osmosis is the diffusion of water only. It's going from high to low, and keep in mind I keep using the word water here because that's the only example you can use. No energy is required. You don't need a protein to do this. There are some proteins that we can use for this if we need to rush water in. Let's say you're not drinking water for a while. We can make proteins so if there are water around, it can suck it into the cell. But really, no membranes required for this one. We did a little bit of this, and when we talk about it, we talk about types of cells. And the different types of cells that experience water differences on one side to the other is called tonicity. But basically what we're talking about is cells being either hypertonic, isotonic, or hypotonic. So basically with hypertonic, what's going to happen is one solution is more concentrated than the other, and there's going to be a movement of water. In isotonic, it's going to be equal concentrations on one side of the cell and the other side of the cell. And hypotonic is one solution is less concentrated than the other, and water will move into the cell, and then eventually what will happen is the cell will explode. Let's look at it. So let's look at a hypertonic solution. A hypertonic solution is where the water is going to be, so the way to do it is, you could see this is the solute concentration. We want to know what the solvent or water concentration is. So that means there's 80% water on the outside and 98, I'm just going to write it over here, 98% inside. So where's there more water? There's more water inside the cell. And because there's more water inside the cell, the water is going to start to rush out of the cell, and eventually what's going to happen is the cell will shrivel. And when the cell shrivels, we're going to have a major problem. So the cell will actually die. We're hoping your body's like this, where it's equal concentrations inside and outside the cell, so your cells are happy. But we could have a situation like this, where the solution outside the cell is higher in water. So here it's going to be like 99.8. And here it's 98. It doesn't sound like a big difference, but it's enough that water wants to go from a high concentration to a low. And because of that, water is rushing into the cell. And because water is rushing into the cell, what we're going to see is a situation where a cell is going to get really big and eventually explode. Where here, we're going to see a happy cell, because this is what it wants to be like. And here, we're going to see a cell eventually shrivel up, and either one of these won't be able to function well. Here you could see a normal cell, isotonic, happy. Water's going in, water's going out. I put this plant one here just because it's a little easier to understand what's going on. So this would be a happy plant or a happy cell. Here in a hypotonic solution, water's rushing in. As water rushes in, eventually what's going to happen is the cell's going to explode. Same thing here. And then here in this hypertonic solution, water is rushing out. And as water starts to rush out, the cell sort of implodes. And here, this is where you can really see it. The cell sort of comes apart, and that's pretty much it. We're only going to be studying animals, but I wanted to show you that just so you can see it. So we've discussed osmosis, and we've discussed diffusion. Another type of movement of high to low is called facultative transport. And facultative transport just means that something was really big or it had a charge. Remember how non-charged items or really small things can move across? Well, let's say this is really big or has a charge. The only way it could come through is if it had a protein. And that's what you're seeing here. So this is facultative diffusion. It's going to still go from high to low till it gets to equilibrium. No energy was required, but we definitely need a protein channel. And we're definitely going to need that lipid bilayer. So we're still going from high to low, but we need that protein channel. And basically what's happening here is that there is a glucose molecule. Only thing that could go through this protein, the special protein, is a glucose molecule. It will go into the cell, and then eventually what happens is it travels to the other side. So it's going from one side of the cell to the other, and then kind of gets spit out. So glucose on one side goes through the little protein, and then eventually gets spit out to the other side. So the only way this could happen is with this protein. But there's one more method that we can use to move stuff into or out of the cell. And it's called active transport. Active transport definitely requires a protein, and it's going to require energy. And the energy that it requires is in the form of ATP. And so this one definitely requires energy, requires a protein channel. It goes against the concentration gradient. So this is the only one that goes in the opposite direction. So all the other ones went from high to low. This one's going to go from low to high. So this is going to be like a pump. And because it's like a pump requires energy, and it's going to move things from the opposite direction of passive transport. So a lot of energy is going to be used. For a cell, it could be that it's moving waste out of the cell, or there's going to be some ways that we're going to move muscles and neurons that we'll talk about in 201 that use this active transport method.